We will now turn to the topic of how to handle negative results, a topic that most of us in research, if not all, have to deal with, and not only once, but most likely repeatedly. Let's first define what we mean by negative or null results. Negative results are any results that do not support or that contradict the original scientific hypothesis. To avoid any confusion, we need to emphasize here that we are talking about the scientific hypothesis, as we, the scientists, would formulate it, in contrast to how statisticians would formulate it. For example, we expect that drug A will slow down the progression of the disease B. This would be our scientific hypothesis, and our studies may turn out not to be supportive or even contrary to this hypothesis if the drug has no impact on disease progression or makes it even worse. These would be a negative or null result from the perspective of our original hypothesis. In this presentation, we will discuss why negative results are unavoidable, what makes negative results unwanted, and how to derive value from negative results. The last point is particularly important. Failures in science are as important as nominal successes. Both positive and negative results are essential for the progression of science. But let's start with the question, why most of us do obtain and will obtain negative results? Let's say we are searching for a novel antipsychotic drug, acting as an antagonist at dopamine D2 receptors. And let's say we have 1,000 drug candidates to evaluate. Nearly all currently used antipsychotic drugs are dopamine D2 antagonists. So we have a lot of knowledge about properties that are essential for a dopamine D2 antagonist to exert antipsychotic effects. To be successful, a molecule should be able to penetrate into the brain, occupy a sufficient percentage of the target receptors, and so forth. If our novel molecule has all such desired properties, the probability of success is high, but still not 100%, because there are often factors that are difficult to control. So let's say for an old and very well-known and extensively validated drug target, we expect to see desired pharmacodynamic effects in 50% of the molecules entering our assessment. Out of 1,000 novel drug candidates, 500 should demonstrate desired effects and 500 should fail. We will power our studies at 80%, so that out of 500 active compounds, we will be able to detect 80%, while 20% will come out as false negatives. And we will set the significance level at 5%, so that from the 500 not active molecules, we will have 95% true negatives and 5% false positives. 80% of the true positives among 500 actives is 400. 5% false positives results in 25 molecules. So there will be 25 false positives among a total of 425 positives, or about 6%. So in this scenario, the false discovery rate will be 6%, which is very low. But how many of us like to work in a field that is so well researched as the one in this scenario? A field where we may be repeating what dozens, if not hundreds of scientists have done already in the past with very similar results. Most of us find it more attractive to work in more challenging fields that have not seen too many positive results. Or better to say, fields with a low probability of a true effect that eventually translates into clinical benefits for patients. In this example of neuroprotective drugs, we set the probability of a true effect at 1%. Out of 1,000 novel drug candidates, 10 should demonstrate desired effects and 990 should fail. We will again power our studies at 80%, so that out of 10 actives, we will be able to detect 8, while 2 will come out as false negatives. And we will set the significance level at 5%, so that out of 990 not active molecules, we should have 95% true negatives and 5% false positives. 80% of 10 actives are 8 true positives. 5% of 990 are 49.5 false positives. So in this scenario, the false discovery rate will be 86%, which is indeed quite high. And now let's make this scenario closer to real life. We have already discussed that the vast majority of preclinical studies are underpowered, and the power rarely exceeds 20%. So what happens if we now change the power from 80% to 20%? The number of true positives goes down from 8 to only 2, and the false discovery rate increases to 96%. Just think about it. The probability of obtaining false positive results is close to 100%. So what can we conclude from these examples? Underpowered studies in the fields with a low prior probability of obtaining a true effect is what makes negative results unavoidable. But then again, we do like to work in fields where prior knowledge is limited. We do like to see our work as being described as a breakthrough, as pioneering or groundbreaking. We do like to describe our findings as surprising or unexpected, don't we? If so, we must be prepared to see negative results. And when we do get and obtain negative results, we often prefer to keep them to ourselves. Indeed, most negative or null results are actually never written up. In this example, 221 studies from a program in social sciences were followed up in order to analyze what kind of results were eventually published and which results were not. It turned out that most strong and positive results were published in scientific journals. In contrast, however, most null results were never even written up and ended up in the so-called file drawer, hidden and unavailable to be considered by other scientists, as shown by the dark green color in the bar on the right side of the diagram. So that brings us to the question, why are negative or null results not published? There are several reasons. For example, I may look incompetent. Maybe there was a technical error or my experiment wasn't done properly. I may look stupid because I've asked the wrong question. I may waste my time. My supervisor doesn't appreciate to publish negative results. It takes so much extra effort to publish negative results. I could publish the interesting or positive work in the meantime. There's a lack of interest. Journals aren't interested in negative results. Negative studies are often poorly cited. If published, then only in a journal with low impact factor. My career might get blocked. You may make enemies, especially if you cannot reproduce data from a famous group. You may struggle to get the next grant. These are all examples of anticipated feedback or answers from our peers or supervisors. But is this really true? There is often a lot of value in negative results, and the certain principles have been developed to help scientists decide when it makes sense to invest efforts despite anticipated negative feedback from our peers or supervisors. A decision can be made in 3 steps. First, one has to answer the question as to whether the observed pattern of results can indeed be presented as negative. Are the results really negative? If the p-value is above the magic level of 0.05, it may not mean anything. And in most cases, it is not sufficient to conclude that the results are negative and can be disseminated as such. It is essential to use adequate data analysis methods to establish the results as statistically negative. Let's discuss a hypothetical situation. We have done a study with 2 groups, control and treatment. So let's call them Group A and Group B for simplicity. And we are looking at the difference between the group means. In the worst case, the difference between groups is equal to 0. In the best case, the difference exceeds a certain threshold that corresponds to a minimum of what would be biologically or therapeutically relevant. There are several possible outcomes of our study. For example, we may see relevant differences between groups A and B, with a 95% confidence intervals of the mean difference not crossing the zero line but covering the threshold for minimally relevant change. An opposite case would be when the confidence intervals do not cover the threshold but cross the zero line. In this case, we say that groups A and B are similar. Another possible outcome is when the groups are not different and not equivalent because the confidence intervals do not cross either the zero or the minimum difference lines. Finally, we may face the following outcome: that the confidence intervals cross both zero and minimum difference lines. This is, in many cases, what we mistake for negative results. However, in this scenario, we are dealing with inconclusive evidence. Let's say that we have done the analysis that supports the conclusion that our results are indeed negative. The next step would be to exclude that this was the result of a technical failure, whether a malfunctioning equipment, an assay not properly set up, or us lacking skills in conducting the study. Next, we make sure that the study was designed and conducted following high research rigor standards. For example, we have already discussed that underpowered studies are at risk of producing biased data. This will also be recognized by reviewers and readers of our publication. This publication, describing the negative results, will have even more weight if it followed recommendations for best reporting practices and if disclosed all information on how the data were collected, if inclusion and exclusion criteria were properly defined, measures against risks of bias, such as blinding and randomization and so forth, implemented and discussed. Finally, in some cases, one may reach out to peers and ask whether they have made similar experiences, whether they have supportive or complementary evidence. This will increase the confidence that these negative results are not just a technical failure and will also increase the potential impact once the results are disseminated. Let's turn to an example of a PD-1 immune checkpoint inhibitor that was reported several years ago to reduce pathology and to improve cognition in mouse models of Alzheimer's disease. As PD-1 inhibitors are already on the market for oncology indications, it may be tempting to rush into conducting a proof of concept in patients with Alzheimer's disease. However, we of course need to be aware of the very low probability of success, high costs, and long duration of clinical trials in the Alzheimer's disease field. With that in mind, it is wise to repeat novel, exciting preclinical findings before engaging in an expensive clinical investigation. So did 3 pharmaceutical companies that all attempted to confirm the originally published efficacy claims of this PD-1 inhibitor. However, as they all came up with negative results, they decided to combine their results. Indeed, when combined, evidence coming from independent laboratories provides much more confidence, and the conclusion published together by the 3 pharma companies was that the animal model data do not support further evaluation of PD-1 checkpoint inhibition as a therapeutic modality for Alzheimer's disease. So, we have given an affirmative answer to the question whether our results are indeed negative. Now, as the last step, we need to find arguments why results are exciting not only for us but also for our peers. A high impact potential is typically what helps to build an argument. Can our data have an impact? Can our data affect decisions such as whether to initiate expensive and long-lasting clinical studies, for example? Let's use another example from the field of Alzheimer's disease. Several years ago, a publication in Science suggested that the drug called bexarotene reduces amyloid A beta pathology and reverses cognitive deficits in mouse models. Very soon after the publication came out, a clinical proof of concept study was initiated. The clinical study had already started before a series of reports appeared in the same journal that all questions the robustness of conclusions in the original report. Even though the clinical study was already ongoing, these publications were nevertheless very important because they contribute now to our understanding of why the clinical study also came out negative. Besides the impact on decisions about clinical trials or the potential impact on policies and common research practices, the dissemination of negative results has another very important function. In many fields, the conduct of research is associated with costs and the use of various resources. Especially in those fields of non-clinical research that involve laboratory animals, we need to take ethical considerations into account. If our negative results are not published, there is a risk that someone else will decide later to test the same exact research hypothesis that we have already tested. So, disseminating our negative results can reduce the waste of ethically sensitive resources. To sum up, we should always be prepared to obtain and see negative or null results. Therefore, we should always plan studies in such a way that if negative, the quality of our research outcomes is certainly high enough to be publishable. As we have already discussed in another lesson of this course, it is strongly recommended to pre-register all important studies and to consider publications in the registered report format, because this novel publication format essentially guarantees the publication of all high-quality results, whether positive or negative. Obtaining and handling negative results is part of the scientific endeavor, and using and considering negative results is an important way of how science progresses. We just need to accept and learn how to turn failures into successes.